Hi, my name is Klaas van der Wijk from Cornell University, and today I'd like to introduce you a new resource for Arabidopsis called Arabidopsis PeptideAtlas. The Arabidopsis PeptideAtlas provides a compendium of results from uniformly reprocessed mass spec proteomics datasets. And what we do is we download datasets from ProteomeXchange and reprocess the raw files through the transproteomic pipeline. A preprint is available here that describes the project by archives. This project is a collaboration between my lab, together with Chi Sun and Sasha Bukarev, as well as Eric Deutsch, Tammy Leppert, Chi Sun, and Luis Mendoza at the Institute for Systems Biology in Seattle. And this project is sponsored by the National Science Foundation. So let's start at the origin. So where do these data come from that we use? They come from ProteomeXchange, and that's a consortium of different databases, and most of you are familiar with Sprite, where you submit your raw mass spec and other information to. When we looked, and this is the statistics for July 2020, we looked what data sets were available for Arabidopsis, and there's 350 data sets. And then we evaluate the different properties, the different features of the data sets. For instance, what instruments were used, were there particular subcellular fractions analyzed, for instance, chloroplast or ER, and what sort of post-translational modifications do these PXD particularly study, and the most popular one being phosphorylation, followed by acetylation and ubiquitination. So to make this peptide atlas and to see what we did, here's the workflow. So we selected, from those 350, we selected 52 data sets and a particular criteria and priorities. We then searched this data using a uniform pipeline, the transproteome pipeline, against ARAPort 11, the default database. If things don't match to ARAPort 11, they were also searched against STAIR 10, small open reading frames, and UniProt and community services, and that's all at a controlled false discovery rate. So what this will result in is that proteins are identified, and we built this chromosome-based peptide atlas. And while we're researching, of course, we also mine these for different metadata. And so what you can find now in the peptide atlas, you can find proteins that are identified using a tiered system. You can look at splice form, non-ARAPort 11 proteins that are identified, small open reading frames, but also N-terminal and phosphorylation acetylation, as well as other PTMs. And collectively, you can see if a protein, how often was it identified, and how was these observations distributed across organs and subcellular localizations. And this data set, or the peptide atlas rather, is integrated now with STAIR and PPDB and also JBrowse. So here's the summary. So we matched 39 million spectra out of 143 million spectra that were searched. And this identified nearly 500,000 of unique peptide and more than 17,000 proteins in ARAPort 11 at the very highest confidence using the particular criterion. There were an additional 3,500 proteins at lower confidence levels. In addition to ARAPort 11, we also have peptides that match to other non-ARAPort 11 proteins, including small open reading frames. This can be used to improve the Arabidopsis genome annotation. And then finally, we particularly focus our attention on phosphorylation as the post-translational modifications. So on the open page, the first page of Peptide Atlas, you can find the statistics. You can actually click, for instance, on the 17,000 proteins identified at this highest confidence tier. You can also look which contaminants we found or non-Arabidopsis proteins. And this is all, you can also look per chromosome. We can see that for each of the 5 chromosomes, 1 nuclear chromosomes, about 22% we have no peptides for these protein coding genes, whereas about 60–65% of the protein coding genes for each of the chromosomes have very high confidence level identifications. So another way to explore this database, to type in a particular protein ID, just this one here. And when you click them, you see a number of pages. You can first see that for this protein, the peptide atlas identified 22 peptides, and there's 198 observations. You can link out to other databases, and here you see the peptides that are matched on the primary sequence, and here you see a different view of that. You can see at the N-terminal region there's no peptides that were matched, suggesting it's probably a cleavable signal peptide. You can you also find here the list of all the peptides. Is there the number of experiments these peptides were identified in, and here you see the sequences. You can also see in which experiments these proteins were identified, and here you can see the PXDs and a little description of what sort of data there were and what sort of instrument and what sort of papers associated. And finally, here you see for all the peptides that are identified for this protein, you see which data sets these peptides were found, and you can see these particular data sets, the peptides that are most abundant. If you click here in the individual peptide, what you see then is you actually have a list of these peptides in their annotated raw spectrum and information of where they are found. And here you see yet again a frequency of the peptide observation for individual experiments. You can see here this peptide was identified in 30 out of, there were 30 observations per million spectra. And finally, you can link back to the original PXD file to read more about it. So if you click here, you go back to ProteomeXchange, and so you can read the original submission and other information that they provide. So, of course, this is only 7 minutes, and if you're interested in more information, you can look at this preview, and this article is currently under review, and subsequent, we are working now on the subsequent PeptideAtlas build, and this will incorporate millions more mass spec data. Thank you very much for your attention.